All right, then I can start. So first of all, I would like to thank everyone who stayed online until now. I am Thomas Evangelidis, live from Prague, and I'm going to talk to you about molecular affinity prediction by artificial intelligence and quantum chemistry. The presentation will be divided in two parts. In the first part, I will describe you deep scaffold algorithm, which is a machine learning based algorithm for affinity ranking. And in the second part, I will describe they can hear me. Okay. So in the second part, I will describe you semi-empirical quantum mechanical methods for entropy calculation. So we usually in drug discovery use the phrase targeting a disease-causing protein. So as an analogy to warfare technology, you can imagine our weapon, which is the small molecule, as a missile, targeting our target, which is a protein, which can be a cell surface receptor, a signal transduction, enzyme, transcription factor, etc. But in all cases, inhibition of its function has therapeutic effect. And actually, in many cases, the 3D structure of this protein is unknown or is solved with a handful of ligands for which we cannot predict the binding affinity of the rest of the molecules we want. However, if we have tested molecules, we can use the current technology to predict binding affinities. So you can imagine deep scaffold as a rocket, a weapon. It has the invariant part, which is the main engine, and the variable part, which is the warhead. There are many alternative warheads in its arsenal, which are simply alternative two-dimensional molecular representations, and each one is specific for a molecular class. For example, it has circular fingerprints or graph convolutional networks, connected subgraphs, pharmacophore fingerprints, variation autoencoders, even LSTM networks. Some of these are specific for macrocycles, for example, others are for covalent inhibitors, others for small fragments, etc. The invariant part, which is the main engine, consists of three components: the train, the select, and the combine. During the train component, multiple, very specific to this problem, multi-layer perceptrons are trained, while in the select component, the best of them are selected to be finally combined into a master network, which gives you the final prediction with small bias, small fluctuations. So you can imagine deep scaffold in operating mode like this. The warhead takes as input the 2D chemical structure and converts it into a bit vector, a vector form. We call it a feature vector. Here I indicate with different colors the output of alternative warheads because multiple warheads can be mounted for optimum performance. Then this is the input, these vectors, the input that goes to the main engine that adapts itself to the target. So it becomes target specific. Once you have the adapted engine, you can feed in the chemical structures of new molecules and get binding affinities. This is a very simplistic representation of what happens. Back in 2017, when I started to program deep scaffolds, I participated more for fun in the D3R challenge, but the results were quite favorable. As you can see here in the overall affinity ranking, I ranked second, while in the free energy set, first. The next year, I participated again with a later version of deep scaffold, and the results were also good. In this year, we were given two data sets. One was for base protease, which contained macrocycles. Macrocycles are very peculiar molecules. And you can see here with blue dots, the best prediction I submitted ranked 13th position, the 13th position. While in the second data set, which was for cathepsin S and contained many molecules, 460, very large data set, I was able to rank first. However, recall that deep scaffold uses only two-dimensional information, while most of the participants, especially those that outperformed deep scaffold in the first set, they used a much wealthier 3D structure information of the receptor. At that time, it was the beginning when I was programming the software. I didn't have many warheads in my disposal. So a few months later, when I finished a new warhead named Neural Graph Fingerprint, which has very good performance with microcycles, I did a retrospective test. I ran deep scaffold, namely again on this data set, and I was able to climb up to the third position. And you can find the code for the Neural Graph Fingerprint on GitHub. It's open source. What I did next was to test deep scaffold on all previous D3R challenge datasets up to 2015. And as you can see here, it has excellent performance. It ranks first in all cases, apart from this one, which ranks at the second position, namely outperformed all the participants at that time. Similar results I obtained when I compared it with the free energy perturbation model from Schrödinger. As you can see here in these histograms, deep scaffold outperforms free FEP in all 8 Protein-Ligand datasets from their publication by obtaining an average improvement in Kendall-Style ranking correlation of the level of 43%. All right, and now I'm gonna dig into some real drug discovery applications very fast. The first one I want to describe you is against CMYC protein, which is an ideal target for deep scaffold because it lacks 3-dimensional structure. As a monomer, CMYC, it doesn't assume any structure, but when it finds its partner, they form a heterodimer. That heterodimer binds to DNA and controls the transcription of hundreds of genes, which are implicated in cell growth, cell proliferation, cell death, and eventually many types of cancer. So it's a very popular target. So what we did here with my collaborators was to take the published inhibitors in the literature, which in our experiments in the lab, they are weak. And I trained a deep scaffold model. With this model, I screened the whole NCI database, and we selected the top 20 molecules for experimental validation. Using SPR, we identified one molecule, M19 indicated here, which had very low dissociation constant. Thus, we measured the profile, the interaction profile of this molecule in NMR. And that allowed me to identify the interacting groups in the molecules. So I created small fragments containing interacting groups. And in the second iteration of drug discovery, I queried the whole NCI database for molecules containing these interacting groups. So using an enhanced deep scaffold model this time, which included the 20 molecules that we tested at the first round, I screened these selected molecules and we ordered the top 10 of them for testing. Among them, we found one, M29, which had very low KD, 2 micromolar. It's almost equal to the dissociation constant of the natural partner of CMYC, which is max. And we measure that by 3 orthogonal techniques, SPR, ITC, and MSD. The next example I want to show you, a real application, is for cathepsin B1 from Schistosoma manzoni, which is a very notorious parasite that causes schistosomiasis. This protein is a cysteine protease and digests our blood proteins in order for the parasite to use it as nutrients. And of course, it's abundant in parasite gut and it's essential for its growth. So what I did here was to take an old publication from 2011, use only 16 vinyl sulfone covalent inhibitors, and with this handful of molecules, I trained deep scaffold using a special warhead. This warhead, I call it QM circular fingerprint because it's the conventional circular fingerprint enhanced with electronic properties of the molecules. So using this warhead and deep scaffold, I ranked 236 vinyl sulfone analogs, and eventually we selected the top 34 for synthesis. Then collaborators tested them in the lab and found that 22 of them were actives, 9 were low or barely active, and 3 were inactive. However, if you calculate the correlation between deep scaffold score and all the compounds, the 34, including the inactives, you get a very high number. This Kendall-Style was 0.6, is the highest I've ever obtained, and this is quite nicely depicted by the p-value, which is at the order of 10 at the -7. It's very low. So to close the first part, Deep scaffold does not require 3D structure, needs minor human input, and you can screen quite structurally diverse compounds. It's extremely fast. It takes about 1 second per molecule. However, you need to have training samples like every machine learning algorithm. The future plan for deep scaffold is to engineer a receptor-specific executable upon request and give it to the user in order to screen super fast, rank super fast, hundreds of molecules. I'm also finishing soon the broader software of deep scaffold, which I dubbed Deep Heat Miner, and it's specific for classification, namely for virtual screening of large chemical libraries when we don't have accurate binding affinities for training. And now let's jump to the entropy part. Entropy has 4 components, but the most important of the components is the conformational entropy. And in order to calculate the conformational entropy, you need to conduct thorough conformational sampling. Thus, the first step of my protocol is conformer generation using Schrödinger tools. But because the OPLS-3 force field from Schrödinger has inaccuracies, it's a molecular mechanics force field, I subject these structures into geometry optimization using a semi-empirical quantum mechanical method. In this case, PM6 and the Cosmo-implicit solvent. So once I have these optimized geometries, I cluster them into microstates, and then using these 2 equations, which are inspired by Gibbs entropy, I estimate a descriptor. I call it conf-S, and this quantifies the conformational entropy of the molecule. Conformational entropy can have several applications. I'm going to show you 3 of them. And the first one is about free energy prediction. This is a real case we had given by our collaborator lab, 1,200 molecules for Mark IV kinase, and we had to classify them into active and inactive. As you can see the histogram at the bottom, these molecules, these sets, contained structures with many rotatable balls and quite flexible. And this means that the conformational entropy contribution to binding is high. So what I did was to select from a public database, decoys, another set from a structurally unrelated protein, a polymerase, which had a similar distribution of rotatable bonds. This means that in this dataset also, the conformational entropy plays a quite important role in binding. And this is very nicely depicted in this ROC curve. With magenta, you see the performance of the enthalpy alone from the semi-empirical quantum mechanical scoring. It's very bad. It's like a random classifier. However, the entropy alone with blue line, it does much better. And this is peculiar. So the ideal case scenario would be to combine these 2 into a single scoring term. I use as a reference in this case GlideSP, which is an excellent software and was the best performing scoring function in this case. And you can see it does quite well for this set. So I combined the enthalpy and the entropy using machine learning in order to surpass Glide. And I created a model which I dubbed ML delta G, which gives you more accurate predictions of the free energy of binding. And you can see with yellow line that it marginally outperforms Glide, which was already good in this case. This was a proof of concept that this scheme works. The second real example I want to show you, we're about to publish this now, is for STING protein. In this case, we had 6 molecules. They were cyclic dinucleotides. 3 of them, the colored green in the table, were doubly fluorinated, while the other 3 were doubly hydroxylated. We know from the experiments from DSF that the fluorinated compounds bind stronger to the protein, while the hydroxylated less strong. However, the enthalpies I calculated from all the snapshots of very large molecular dynamics trajectories, they were misleading. They failed to rank them correctly. In contrast, the same didn't happen when I calculated the entropy descriptor on the DFT level of theory. As you can see here, the green molecules, the fluorinated, they have less entropy than conformational entropy than the hydroxylated. And this is very nicely depicted in the figures below. The first row shows the fluorinated molecules, which assume less conformation than the hydroxylated molecules at the bottom row. The final application I want to show you before I close is how to combine this entropy descriptor with machine learning. In this example, I'll show you the circular fingerprint which is expressed as a bit string. So in order to incorporate to that bit string the conformational entropy, we need first to discretize it into an integer number, and then convert this number into a bit vector. Then we just append a bit vector with a bit string to have an enhanced vector for string. I tested this in map 4K4 from 2015. As you can see with blue dot, that's the performance of deep scaffold using the original vector. And with this symbol, I show you the enhanced vector with the entropy. It's much higher, not much higher, it's significantly higher. So to conclude, enthalpy alone is insufficient for scoring, but we can calculate the entropy using quantum mechanics and combine it with machine learning to get more accurate description of the free energy. On the other hand, we can use the entropy alone to improve ligand-based scoring with machine learning. And currently I'm generating and created a general case machine learning model to predict the free energy of binding using entropy and enthalpy. So I would like to thank you. Deep scaffold is solely my creation, and I will launch a company soon. While quantum mechanics research, I do it in the group of Professor Pavel Hobza. And these are the collaborators from other cases that I showed you. Now we can move to the questions if there are any.